The making of a scientist. We all know this chapter is very complex and difficult. But don't worry. I'm here to help you. This is a story about a curious child who became a scientist due to that curiosity. You know, this chapter is to be remembered, as it's not much interesting. Don't worry. I bet you will know this chapter in this video. If you watch it with focus, and reading concentrated. The captions you see here. An only child, Bright grew up north of Reading, Pennsylvania. There wasn't much he could do there. He certainly couldn't play football or baseball with a team of one. There was one thing he could do there. Collect things. So he did, and did he ever. Beginning in kindergarten, Bright collected butterflies with the same determination that has marked all his activities. He also collected rocks, fossils, and coins. He became an eager astronomer, too, sometimes stargazing all night. He had a driving curiosity along with a bright mind. He was also encouraged by his mother and his interest in learning. She took him on trips, bought him telescopes, microscopes, cameras, mounting materials, and other equipment, and helped him in many other ways. His mother said,
"I was his only companion until he started school. After that, I would bring home friends for him. But at night, we just did things together. He was my whole life after. His father died when Richie was in 3rd grade. We spent almost every evening at the dining room table. If he didn't have things to do, I found work for him. Not physical work, but learning things. He liked it. He wanted to learn. And learn he did. He earned top grades in school. On everyday things, he was just like every other kid."
By the time he was in 2nd grade, Bright collected all 25 species of butterflies found around his town.
Remember, Bright had collected 25 species of butterflies found around his town, in his 2nd grade.
That would have been the end of his butterfly collecting. But then, his mother brought him a children's book called The Travels of Monarch 10. That book, which told how monarch butterflies migrate to Central America, opened the world of science to the eager young collector. At the end of the book, readers were invited to help study butterfly migrations. They were asked to tag butterflies for the research by Dr. Frederick Urquhart of the University of Toronto, Canada. Anyone who found a tagged butterfly was asked to send the tag to Dr. Urquhart.
Remember, Bright's mother gave him a book called The Travels of Monarch X. That book told how butterflies migrate to Central America. At the end of the book, readers were invited to help study butterfly migrations. They were asked to tag butterflies for research by Dr. Frederick Urquhart of the University of Toronto, Canada. Anyone who found a tagged butterfly was asked to send the tag to Dr. Urquhart.
And soon, Bright was attaching tags to the wings of monarchs. Oh, I am tired of narrating. Now see Richard Bright himself. The butterfly collecting season lasts only 6 weeks in late summer, around Reading. If I am going to chase them one by one, I will not catch many. So, my next step is to raise a flock of butterflies.
Remember, the butterfly collecting season lasts only 6 weeks in late summer, around Reading. So Bright decided to raise a flock of butterflies.
Do you know that he raised butterflies? He would catch a monarch butterfly, take her eggs, and raise them in his basement through his life cycle. From egg to caterpillars, caterpillars to pupa, and pupa to adult butterfly. Then he would tag the butterflies, and let them go. For several years, his basement was home to thousands of monarchs in different stages of development. Now, I am losing interest in tagging butterflies. It's tedious and there's not much feedback. Only 2 butterflies, that I have tagged, are recaptured. And they were found not much far from my home. In the 7th grade, he entered a science fair. His entry was slides of frog tissues, which he showed under a microscope. But he had lost. Remember, in 7th grade, he entered science fair, but lost. Because he had neatly showed frog slides under a microscope, and not done any real experiment. It's really a sad experience to sit here and not get anything, while everyone has won something. Now I have realized that the winners had tried to do real experiments, not something made a neat display. Now, I know, I will have to do a real experiment for next year's fair. The subject I know most is the insect work I've been doing for past several years. So he wrote to Dr. Urquhart for ideas, and came back many suggestions for experiments. Those kept him busy all through his high school. For his 8th grade, Bright tried to find the cause of a viral disease that kills nearly all monarch caterpillars every few years. He thought that the disease might be carried by a beetle, so he tried raising caterpillars in the presence of beetles. He didn't get any real results, but he went ahead and showed he had tried. This time, he won.
Remember, in his 8th grade, he tried to find the cause of disease responsible to death of many monarch caterpillars. He didn't get results, but he won. He had tried.
Next year, his science fair project was testing the theory that viceroy butterflies copy monarchs. The theory was that viceroys look like monarchs because monarchs don't taste good. Viceroys, on the other hand, taste good to birds. So, the more they look like monarchs, the less they are to become the dinner of birds. Remember, in the next year, his science fair project was testing that viceroys copy monarch. Viceroys look like monarchs because monarchs do not taste good to birds. His project was to see whether birds would eat monarchs. He found that a starling, a common European bird, wouldn't eat ordinary bird food. It would eat all the monarchs it could get. This project was placed first in the zoology division, and overall third in the county science fair. In his 2nd year in high school, he began the research that led to his discovery of an unknown insect hormone. Indirectly, it also led to his new theory on the life of cells. I studied the purpose of the 12 tiny gold spots present on the non-erect pupa. I think that tiny spots are not just ornamental, but they produce hormones necessary for their development into butterfly.
Remember, in his 2nd year, he began the research of an unknown insect hormone that led to his discovery to his new theory on the life of cells. He studied the 12 tiny gold spots present on the monarch pupa. He concluded they were important for the development of the pupa. This project led him towards his new theory on the life of cells.
The project won him first place in the county fair, and entry into the International Science and Engineering Fair. There he won 3rd place in zoology. As a high school junior, he continued his advanced experiments on the monarch pupa. There too, he won 1st place at the International Science Fair and gave him another chance to work in the Army lab. Now, listen carefully. In his junior year, he was looking at X-ray photos of the chemical structure of a hormone. Then, he got the idea for his new theory about cell life. I found a possible answer to the puzzle: how the cell can read the blueprints of its DNA. Me and my college roommate, James R. Wong, we both drew pictures and constructed plastic models of molecules to show how it could happen.
Remember,
during his junior year, Bright got the idea for his new theory about cell life. It came when he was seeing X-ray photos of chemical structure of a hormone. He found a possible answer to the puzzle: how the cell can read the blueprint of its DNA. Bright and his college roommate, James R. Wong, drew pictures and constructed plastic models of molecules to show how it could happen. Together, they both later wrote a paper that explained this theory. At the age of 22, Richard Bright excited the scientific world with a new theory on how cells work. They both explained an article in the Proceedings of the National Academy of Science. Remember, at the age of 22, Richard Bright excited the world of science with a new theory on how cells work. He and his college roommate explained the theory in an article published in the Proceedings of the National Academy of Science. Don't go anywhere. It's not ended yet. Now, watch a small summary that will be even more helpful for your exams. Ready, steady.